This is Bernard Paulson. This lecture is the first of many that cover chapters in the book pictured on this slide. This book teaches how one builds dynamic simulators of biological networks. The first lecture covers a few topics that serve to introduce this field and this undertaking. First, we'll describe the systems biology paradigm that has arisen relatively recently. Then we'll describe briefly what a biological network is and perhaps make some comments more broadly about networks. We will then talk about the motivation for building mathematical models. We will then introduce the key concepts that underlie the analysis of dynamic states. We will then talk about the different approaches to building models. And finally, in the last part of the lecture, we will talk about the sources of the data or the information that goes into building biological models and show that this data can be organized in the form of two key matrices. First, we'll talk about systems biology. It is important to note that the first genomes were sequenced in the mid-1990s, and as a result of that, systems biology arose. It can be considered as a genome-enabled science or a post-genome science because genome sequences allow us to obtain a full or comprehensive view of the genome complement of the target organism. The systems biology paradigm is broken down into 4 steps. First is the elucidation, understanding, and measurement of the components of a cell. These are its chemical components. Then is the reconstruction of the interactions of all these components into networks. The network description can then be converted into a computational model, and that computational model can then be used to compute biological functions that we know as phenotypes. Pictorially, this paradigm can be detailed further as shown in this image. First, there are many types of data that are now available that enumerate biological components. Secondly, the reconstruction process that follows results in a network. At level 2 in this diagram, there is a single bubble that indicates the fact that there is only one reaction network of a particular type operating in a target organism. Then we have the conversion of the reconstructed network into a mathematical format, and that allows us to do a number of computations. The computations are then used in a prospective manner to address several practical applications, such as those indicated at the bottom of this diagram. In this class, we will focus on the construction of dynamic simulators of network functions. In doing so, we will talk a little bit about the topological constraints that networks operate under. This 4-step paradigm underlies the field of systems biology. The function, the role of this class is not to teach you how to obtain data or reconstruct networks, but to take an existing network and build a dynamic simulator of it. I want to say a few things about biological networks to get us started. So what is a biological network? Fundamentally, a biological network is based on a series of chemical transformations. All cellular events ultimately have a chemical basis, and chemical transformations can either involve genuine chemical reactions, that is, the breaking and forming of covalent bonds, or it can involve the association of molecules through hydrogen bonding, for instance, into states into a bound state where two molecules bound together will have a different function than separately. Networks in general, all networks, are comprised of what we think of as nodes and links. So nodes are linked together. In the networks we will look at, the nodes are chemical compounds, and the links between these compounds are chemical transformations. The function of a network as a whole is biological or physiological. So therefore, what's called the functional state of a network is very important because it is related directly to biological functions of the organisms that we are interested in. At the lower part of this slide, we show examples of three commonly studied biological networks. Metabolism, which is universal to all cells, involves transformation of small molecules, the consumption of substrates, and the formation of metabolites such as amino acids that go into protein synthesis. The second network pictured is that of transcriptional regulation. This kind of network involves many regulatory protein and functional protein that bind to DNA to control the transcription of the information found on it. The third network shown in this picture is a signaling network, and this is a network that acts in tissues where different cell types send signaling molecules to communicate with one another. In this class, we will primarily focus on a metabolic network. It is important to note, though, that these are not three separate networks functioning inside cells. There are many compounds that are found in all of them. For instance, cyclic AMP is a metabolite. It's also a compound that may bind to a transcription factor that is involved in regulation of gene expression, or it could be found in a signaling network. So it's important to realize that these networks pictured here really form a single larger network in addition to other cellular functions. Building a network on a genome scale is a laborious process that is now executed under well-defined workflows. A bottom-up network reconstruction represents a plethora of biochemical, genetic, and genomic information in addition to other types of information not shown here. Such a network reconstruction is really a multi-scale knowledge base as shown in this slide. It will have chemical structures in it, it will have genetic information in it, it will have chemical reactions in it, it'll have pathways in it, it'll have the information about the entire set of metabolic capabilities encoded on a particular genome. Often, there is also information included on the cellular location in which reactions takes place. The network pictured here is the global metabolic network encoded on the human genome that was built over 2 years by 7 young, brave people pictured at the bottom of the slide. As I stated earlier, networks are fundamentally comprised of nodes and links. In our case, the links will be chemical reactions. However, in biology, we don't always have detailed chemical information about the linkage between events in biology, be they chemical or cellular. Therefore, there is typically a spectrum of depth of knowledge available about links in biological networks. They go from being fuzzy or abstract to the very specific. On the abstract end of the scale, we may think about risk factors, for instance, in human disease. Cholesterol levels are risk factors in heart disease, for instance. The mechanistic relationship may not be well known, but these two events are somehow linked together or correlated. That would be a very non-specific information. At the other end of the spectrum, we have highly specific and mechanistic information about how two objects are linked together in a biological network. This class will operate on this end of the spectrum, where we will have detailed chemical, kinetic, and thermodynamic information about the links that we want to describe. We will primarily focus here on metabolic processes and include in them the protein that drive these processes and how metabolites bind to them. We all know in this day and age that we have a vast amount of information available to us through the internet. Biological information is no exception, and this slide shows just a small example of websites from which one can get biological information about metabolites, protein, regulatory events, organisms, and so forth. The websites given in this slide are of particular use to this class. The last column in this table is important, not in detail, but in concept, as it indicates the level of curation of the information found on that website. And this relates to how well the information has been quality controlled or quality assured. For some organisms and some processes, we have very detailed information available that is heavily curated. For others, we are not so lucky. Before beginning to look at and admiring network maps, it is important that we all keep the biophysical picture of cells in our minds. Cells are three-dimensional objects, very crowded, very filled with different molecules. These molecules are moving around by thermal motion or by deliberate motion within inside the cell. They look nothing like a circuit, electronic circuit, a traffic map of a city, or the distribution map at a retailer, or the routing map, say, of an airline. These are all networks and are described by similar mathematics, but the physical embodiment is very different. This particular slide or image shows a reconstruction of the entire E. coli cell. What is pictured in here are all the macromolecules, the membranes, the DNA, the flagella, and so forth to scale to show what the biophysical picture of a cell is. This is multi-scale, and one can zoom in and look at the finer details of a picture like this one. And this enlarged view shows the flagella, how it's hooked into the cell wall and the membrane of E. coli. Shows a little bit more detail of the nuclear region or the nucleoid. Shows some ribosomes and some protein. And one could zoom in further and look at the finer detailed structure of a cell. So we should all keep in mind that a cell is a highly concentrated space of various macromolecules that are moving around, reacting, and carrying out different cellular functions. If this image fascinates you, I recommend that you check out the books that David Goodsell has prepared that are filled with beautiful images such as this one. Now let's move on closer to the topic of this book and this class. Let's talk about first the impetus for building models. Why do we build mathematical models? What motivates us to do this? This motivation is captured in a well-known article written by the late J. Bailey. He breaks the motivation for building models into 5 categories. First, to build a model, you have to organize a lot of different types of information into a coherent whole. The network reconstruction process that I mentioned earlier is an example of this. The next thing is the ability to take such information and quantitatively relate it to one another, to systematically look at all interactions at the same time and be able to compute or calculate the consequences of all those interactions operating at the same time. The class will be very heavily focused on this issue. Once you have a model, you can discover various new strategies. You can carry out a number of data mapping procedures onto a model, as an example. When you build models, you often discover that even if the model contains everything that you know about the process, that the model doesn't compute the real biological behavior correctly. This can mean 2 things. Either something is missing in the model and you have to discover missing components, or there are processes not accounted for in the model that are operating. So discrepancy between a model and actual biological behavior is very informative. Finally, we use models to understand essential qualitative and semi-quantitative features of complex processes. For the purposes of this class, we could think about the adaptation of a bacteria to a new growth environment, a pathogenesis, the process of pathogenesis, process of development, and so forth. So there are many reasons for putting models together. Perhaps the most striking and simplest way of putting a motivation for building a model is this quote to Richard Feynman that he left on his blackboard as he passed away. And it states, "What I cannot create, I do not understand." So a model is created from a lot of different sources of information and used to develop an understanding of the process that you're studying. From a practical standpoint, an engineering standpoint, a utility standpoint, models are used to describe complex systems. Certainly in biology, cells are very complicated, comprised of thousands of molecules that come together to form biological function. And these processes cannot be comprehended easily by the human mind. In fact, there are studies that show that the human mind can keep track of up to 5 objects simultaneously and look at their interrelationships. Apparently, chess champions can look at more than that, maybe 7, 8. But once you start looking at dozens of variables at the same time, there is just no way that we can create a mental image of a process of that complexity and estimate how it unfolds in time or how it functions generally. Models are of course prevalent in all engineering disciplines, and there are models now of traffic networks in cities, cell phone networks, airline trafficking, and so forth. Trafficking of airlines, I should say, amongst the different, say, airports in the United States and internationally, of course. The models we are going to talk about here analyze dynamic function. This means change in time. There are, of course, many other types of models, but the ones we are focusing on here deal with dynamic phenomena. Before we begin, I just want to make a few comments about the multi-scale nature of biology. There are sizes of objects to worry about. There is a rate of change to worry about. There's an abundance of objects to worry about. If you look at the first part of this image that gives us a size spectrum, it is important to understand the relative sizes of different biological objects. This the scale on this image is given in meters, so 10 to the minus 9th is a nanometer, 10 to the minus 10th would be an angstrom, and that's the length scale of molecules. So molecules like water, glucose, metabolites are on this size scale. Cellular components, structural components are a little bigger. Membrane thickness, for instance, by lipid layer is on the order of 7 nanometers. The ribosome that builds protein is on the order of 20 nanometers in size. Retroviruses or enveloped viruses like HIV may be 50 to 100 nanometers. A bacterial cell will be a micron in dimensions. The image I showed you earlier of a swimming E. coli cell shows a short rod, if you will, that is maybe 4 microns in length and maybe a micron and a half or 2 in diameter. Eukaryotic cells like yeast will be a little bigger, 5 to 7 nanometers. And then mammalian cells may be up to 20 microns in diameters. I think I misspoke. Yeast cell, of course, is a few microns in size. This length scale spectrum here spans that of cell and molecular biology. The rate at which processes happen is also very, very important. And some of those are indicated in the middle section of this diagram, the green swath, the green strip, excuse me. This is scaled in seconds. So 10 to the 0 would be 1 second. We see that there are many processes faster than that. Metabolites diffuse typically very quickly in cells. The polymerases like RNA polymerase will maybe put up to 20, 30 nucleotides into a growing RNA molecule per second. If you look further up to the right on this scale, 10 to the third, 1,000 seconds. So an hour is 3,600 seconds. So the doubling times of bacteria may be on that magnitude. So E. coli can double as fast as every 20 minutes, which is under very unusual conditions. Typically doubles every 45 minutes to an hour in rich media. More complicated organisms will divide slower. Finally, the blue swath is somewhat important. That indicates the concentration or the abundance of different cellular components. Remember, the nodes in a network are comprised of those components, and it's important for us to know how many objects, how many copies of a particular object are found in a node. So we can look maybe at some of the items on this slide. First 10 to the minus ninth, this is molar, so molecules per liter, moles per liter. If you look at the volume of one E. coli cell, which is on the order of a cubic micron, 10 to the ninth molar would correspond to about 1 molecule per cell. If you move a little bit further up this scale, there are typically 5,000 to maybe 20,000 ribosomes in E. coli. 10 to the minus third on this scale would be millimolar. A few metabolites are in the millimolar range, like ATP is maybe a few millimolar. And the total concentration of metabolites, all of them summed together, may be as much as 300 millimolar in a cell. You may look up the reference given at the bottom of the slide, as it has many interesting facts, numerical facts about cells. It's only 2 pages, this reference, and it's easy to read. So these are some attributes of networks. Multiple size scales, multiple length scales, multiple abundance scales. Networks have states. We are very interested in the state of a network. We are interested in steady states. We are interested in dynamic states. These are often called transient states as they are changing with time. Those may be mathematical definitions of states, being steady or dynamic. Biologically, we think about homeostatic states, we think about physiological states, and it's important to note that these biological states are created by the functional state of the underlying biological networks. For dynamical analysis, there are a few fundamental concepts that we need to become familiar with. I'll just mention them briefly here and detail them in the next lecture. We need to be familiar with the concept of a time constant. So that is the time duration over which a significant change in a process takes place. A half-life is an example of that. For instance, the half-life of the HIV virus is about 7 hours. So in 7 hours, half of the virus inactivates. The second concept is is the notion of aggregating variables together or lumping them into pools. This is a result of a multi-timescale analysis. As different events relax on different timescales, the dimensionality, the dynamic dimensionality of mathematical description shrinks, and that reduces the number of independent variables, leading us to pool variables together systematically over timescales. The third concept here is the nature of the transition from one steady state to the next. And then finally, graphically representing the solution is very important, because if you try to do it numerically, we are overwhelmed with a large amount of different numbers. So graphical representation and interpretation of a dynamic state is very important. The existence of multiple scales, time scales, is very important. We talk about multi-scale analysis or hierarchical analysis. And the image shown in this slide is a glimpse of that. To the right, we show a series of events in words on different timescales. The images to the left of that are different network images tilted to the side to show the aggregation phenomena that are indicated by the dotted blue lines. And at the very bottom, we show the results of dynamical aggregation of variables onto the slowest time scale for a glycolytic model. The slowest time scale in this case, the dynamics of the individual metabolites in glycolysis become irrelevant, and they all move as a group or as a pool. And in the bottom image there, we have drawn a red box around all of these metabolites and simply summed them up. And the size of that pool is just moved by hexokinase. So we have 2 unit fluxes of hexokinase going into this pool, and then ATPase designating a flux out of that pool. So that's a highly lumped description of a very detailed biochemical process at a slow time scale. This description may be a little difficult to follow at this point, but as the class develops, you will gain an understanding of such hierarchical thinking. Now, how do we build dynamic models? There are 2 fundamental approaches in building and dealing with dynamic models. The first one is through simulation. This is what we will teach in this class. The dynamic simulation process is comprised of 5 steps outlined here. First, we have to learn how to formulate the governing equations. That is normally done using well-defined assumptions. We then have to specify numerical values for the parameters that are found in these equations. We then have to specify the condition under which we want to carry out the simulation. So every simulation is a case study. We then solve the equations under those conditions numerically, and this is done by using readily available software packages such as MATLAB or Mathematica. And then finally, we graph the results and analyze them. This 5-step process will be detailed in this course. The second way of building and analyzing and understanding dynamical models is through a deeper mathematical analysis. Such analysis is typically global, meaning it will deal with all aspects of the properties of a model, in contrast to a simulation that is just a case study. We will not describe this material in this class. It tends to be more mathematically involved and require a little more advanced knowledge and understanding. So this approach is typically delegated to a subsequent class. Simulation, in contrast, is practical, relatively easy to implement and understand on a case-by-case basis. The first step for us is to formulate the equations that govern the description, the mathematical description of a biological network. In doing so, we apply a few well understood assumptions that are described in this slide. First, we do the continuum assumption. That is, we are not dealing with individual molecules and their motion, but we treat the medium as a continuum. This is, of course, what all analysis in fluid mechanics, heat transfer, mass transfer, and so forth is based on. We make an assumption that the volume we are looking at is constant. This is perhaps the most concerning assumption we make in building dynamical models of biological networks. We'll talk more about it in the next lecture. Dynamical models in biology are typically based on mass action kinetics and kinetics only, and they often ignore important factors like charge and osmotic pressure. We will not deal with those factors in this class, but these are often important and one needs to keep them in mind. The finer structure of cells is typically ignored, and this relates to the first assumption outlined on this page or this slide. And we typically treat systems as being isothermal, and of course this normally refers to the human body at 37 degrees or the temperature controlled environment in a fermenter or in a bioprocess. So normally we do not consider the temperature effect on kinetic behavior. This effect can be substantial as most kinetic constants change notably with temperature, often described by Arrhenius' law. We also typically treat systems being under atmospheric pressure. Under these assumptions, we formulate what are called the mass balance equations. These equations are the master equations that we use to describe the function of a biological network in mathematical terms. So the way this is done is as follows. We look at every node in the network, which is a compound designated here by X sub I, and all the links or the reactions all the links in and out of that node, which are the reactions that compound participates in. In this case, there are two reactions that form X sub I, designated by V sub 1 and V sub 2, and there are three reactions that degrade X sub I. The V's are so-called fluxes. They will be amount of change per volume per time. The node content is given in terms of concentration, the amount per volume. We'll talk more about these units in a subsequent lecture. The dynamic mass balance that is formed for this compound is shown at the bottom of the slide. So the time derivative of X sub I, so this will be the time change of the concentration of X sub I, is equal to the flow into this node, or the reactions of formation, minus the flows out of the node, which are the reactions degrading X sub I. In a steady state, the inflows and the outflows balance perfectly, and the time derivative is zero. We are going to use quite a bit of linear algebra in this course, so I want to point out that this summation of these fluxes in and out of a node can be represented as an inner product of two vectors. These are shown at the very bottom of the slide. The vector to your right is the so-called flux vector, which is just a list of all the fluxes in a network, and the vector in front tells us whether these fluxes are flowing in or out of a particular node, such as that for x sub I in this case. This is an inner product of two vectors, and that subscript t stands for transpose, meaning that the flux vector is normally a column vector but has been transposed into a row vector in this particular equation. Now, the next thing I want to describe are the workflows that we go through. This slide shows the workflow associated with the two approaches to studying dynamical models, simulation and analysis. They both have in common the formation of the equations. So the equations are shown in this box over here, and this is a matrix equation that we'll introduce in detail later, but it's the time derivative of now a vector of concentrations, and that's equal to a matrix S, that is the stoichiometric matrix, times the vector of fluxes or reaction rates. Those reaction rates, as we'll detail in the next lecture, are functions of the concentrations of the compounds that participate in those reactions, and those rate laws, as we call them, contain kinetic parameters designated here by lowercase k, and these are the kinetic constants. To formulate these equations, we need data and observations. We need basic physical-chemical principles, such as mass action. Then we need to define our problem, which is based on the size of the network we're looking at, the cell that the network operates in, or the organelle it operates in, and so forth. This class will go down the left-hand side of this diagram. As you go through the simulation process, we have to specify the numerical values of the rate constants. We have to specify the conditions under which we are doing the simulation. We numerically solve for the concentrations and how they change over time, and that's designated by x sub t, x as a function of t, time. And then the fifth step, the final step, is to graph the solution or post-process it in various ways. So we will go through the boxes in this workflow that are highlighted in red. The structure of the textbook that we will use is shown in this diagram. It proceeds from the simple and the conceptual to the detailed, the biological, and the relevant. So part one of the book covers the basic mathematics and shows how they are applied to simple conceptual models, often called toy models. Part two shows some of the generic features of biological networks. Part three deals with very specific metabolic processes in a cell. We will build dynamic models of the basic biochemical pathways found in all cells. And part four will show how small metabolites interact with protein to produce realistic biological networks. We will go through all of these parts in this class, and the class comes with a term project that focuses on part four, where you will actually build realistic simulators of real cellular phenomena. The last topic of this introductory lecture is focused on the basic information that goes into dynamic models and how that information is represented in a matrix format. This slide shows schematically the information conveyed in three different matrices. The first one is the stoichiometric matrix called S, and it gives us simply the structure of a network, or its topology, as it's called. So the diagram to the right of S is a connectivity map, shows nodes and links between them. The matrix G, or the gradient matrix, has the kinetic constants in it. And that information tells you how quickly the different links in a network react. What is their characteristic time constant? J is a Jacobian matrix that gives us the dynamic properties of a network as a whole. It is not independent of S and G, but rather it is the matrix product of S and G. The Jacobian matrix is a key matrix in performing analysis of biological networks. We will therefore not talk more about the Jacobian matrix in this class. Finally, this slide shows the many and important attributes of these two matrices. This slide is somewhat important, so I'm going to describe it in some detail. It is absolutely remarkable how much information is contained in these two matrices simultaneously. First, let's talk about their informatic origin. Where do they come from? The stoichiometric matrix comes from information embedded in the genome sequence. Genome sequence gets annotated with the genes that are found on it and their function. Detailed information about the function of a gene product is found in the literature that is now called the bibliome in its totality. So if you're building a stoichiometric matrix describing a network in a target organism, you need to get the genomic information. The matrix G, on the other hand, has kinetic data in it. It has not been built yet on a genome scale, but that could be achieved once we are able to get high-resolution metabolomic and flossomic data. The physical-chemical basis for these matrices is also well understood. The stoichiometric matrix represents chemistry, certain conservations like on elements and charge. The matrix G has kinetic information in it and thermodynamic information in it. Both matrices have important biological features. So S, like I said, is the characteristic of a genome, and it represents a species. So every member of the species would have the same stoichiometric matrix. All E. coli cells in a culture you're growing will have the same stoichiometric matrix. On the other hand, there are sequence variations in those genes that are particular to an individual, and that gets reflected in different kinetic constants, or the different response times of the links in the network. So G is actually specific to an individual. So if we want to look at differences from one individual to another in a population, we need to get G for the different individuals. The matrix S has been found to be useful in studying differences between species because it represents a species by itself, and has also been useful in studying what's known as distal causation in biology, how genomes change over generations. For instance, horizontal gene transfer into a cell may create an entirely new uh, column in the stoichiometric matrix. G is very important in calculating what's called proximal causation, or the reaction of a cell to a stimulus without any change in the genetic material. This includes, of course, dynamic responses that we are interested in. These two matrices have important mathematical characteristics. S is comprised of integers only and is therefore a knowable matrix. There are no errors in integers. On the other hand, G has real numbers in it, and those real numbers will have errors in them. They will have an imprecision associated with them. We will never know the elements of G precisely. From a system standpoint, S has a lot of information about network structure and pooling that we'll talk about later. G will have information about the response times of the links, and therefore has information about the timescale spectrum found in a network, and therefore will be very important in deciphering the dynamic function of a network. Finally, from a numerical standpoint, it's important to point out that these matrices are sparse. That means that very few of the entries are non-zero. For large networks, maybe less than 1% of the elements in a matrix will have a numerical value, the rest are zero. S has stoichiometric coefficients. They are all on the same order of magnitude, and therefore that matrix is called well-conditioned. It's easy to compute with, and is described as being non-stiff. The gradient matrix G, on the other hand, may have elements that differ by many orders of magnitude, sometimes 6, 7, 8, 9 orders of magnitude. This makes the matrix ill-conditioned. It's hard to compute with it because you will have large and small numbers in it, and this leads to what's called stiffness in computations. This slows the computations down. So this is an important slide because it gives you the data and the informatic basis for these two matrices. They contain a lot of different information, and therefore, as simple and relatively empty matrices represent a lot of knowledge. We will now summarize the basic points, the basic issues brought up in this lecture. First, the relatively recent process of systems biology has 4 basic steps: component enumeration, network reconstruction, conversion of networks into mathematical models, and then computing phenotypic functions. As we'll see towards the end of this class, this actually allows us to build mechanistic genotype-phenotype relationships because many of the entries in the mathematical description have a genetic basis, and the computations that we perform represent phenotypic states. In the genome era, or the post-genome era, we now can build biological networks on a large scale. This is a laborious process, and it's not going to be taught in this class, but students need to be aware of the fact that it is now possible to build such networks. The dynamic states of the networks that we're going to study are characterized by time constants, reduction in dimensionality with time that leads to aggregation of variables, and they have very characteristic transitions from one steady state to the next that will be detailed in a subsequent lecture. Dynamic analysis is a mathematical subject. It will come down to dealing with high-dimensional descriptions, multiple timescales, and so forth. And you will need to know what you learned in your ordinary differential equations class and your algebra class. So please go and review those in detail, review those classes in detail, because you will need to understand these two types of mathematics. We will primarily in this class simulate responses, which as I stated earlier, are case studies, and not pursue analysis of equations, which is a more advanced subject. I did mention that there are some assumptions that underlie the formulation of the dynamic mass balances that you need to keep in mind, because sometimes you need to be able to evaluate whether the model that you're using is applicable. If it violates some of the assumptions made, it may no longer be applicable. And finally, in the last segment of the talk, I outlined the contents of two important matrices, the stoichiometric matrix and the gradient matrix, and their many attributes. You will become familiar with these as the class moves along. So this concludes the introductory lecture for this class. The next lecture that follows will focus on some of the fundamentals of kinetics and kinetic analysis. With that in hand, we will then go on to build progressively more complicated dynamic models of biological networks. Thank you for your attention.